Coming to activity 6.1. Here we have to take a potted plant with variegated leaves, meaning एक leaf जिसके अंदर patches होते हैं green color के and yellow color के, right? We can take a money plant also, croton leaves also. The steps that we are going to follow is, first of all, we are keeping it in a dark room and duration is 3 days. Okay, so as to de-starch it. Number 2, we are bringing it in sunlight for 6 hours. Then we pluck a leaf from the plant and we trace it on a paper, mark the green labelings, and further moving to the third step. We are going to give it a dip in boiling water. What happens when we dip it in a boiling water? Obviously it will kill the leaf, right? Then immerse it in a beaker which contains alcohol. Here we have to place the beaker carefully in water bath and heat till the alcohol begins to boil. Alright. Now, there are certain questions followed up, that what will happen to the color of the leaf? Okay, then what is the color of the solution in which it is placed? If we dip the leaf, this particular one, after this procedure, if we dip this in a iodine solution for few minutes, what will happen? Then compare the color of the leaf with the tracing that you have done, where we have traced the green portion and the yellow portion, right? That way. This is the green one I told you, and this is the yellow one. Then, what can we conclude for the presence of starch in various areas of the leaf? For all those questions, first of all, when we take up this procedure, it will decolorize and the solution will turn light pale and yellow and green in color. Iodine solution, when we dip it in iodine solution, obviously the part of the leaf which is green in color and performs photosynthesis will only show starch, and that will surely give the iodine test. Rest this yellow region will not perform the iodine test. So after the activity we conclude that the leaf will be seen in two types of patches, the blue-black patches and the yellow patches. Blue-black are the areas which were earlier green in color and performed photosynthesis, so had starch in it. And the yellow one, because there is no starch, so automatically it will not perform any photosynthesis and no iodine test at all for it. Okay, so the starch that we are talking about here is all because of the chlorophyll which is present in the green region. So only chlorophyll produce starch which is stored in the plant itself. This is first activity. Moving to second activity, activity 2. Here we have to take 2 potted plants and we have to keep them individually, 2 different inverted jars in a dark room for 3 days. Basically क्या करना है हमें? सबसे पहले हम plants लेंगे, same plants. Alright, we have to place these 2 plants in dark room for 3 days. After 3 days of their procedure, we will now place each plant in separate glass plates. Watch glass, one of the watch glass will contain, now these are the 2 glass plates onto which we have kept our plants. Right, here we say that one of the watch glass has KOH pellets in it. KOH means potassium hydroxide. We are keeping it by the side of one of the plant. This is used to absorb CO2. Okay, this is our setup A and this is our setup B. Now this KOH is used to absorb carbon dioxide. We are going to cover both the plants with separate bell jars. Now we will use Vaseline to seal the bottom of the jars so that the setup is airtight. ठीक है. अब इन प्लांट्स को हमने सबसे पहले क्या किया था, 3 days के लिए डार्क में रखा था. नाउ आफ्टर दिस सेटअप वी आर गोइंग टू कीप दीज टू plants in sunlight for about 2 hours. After that, what are we going to do is we are going to take a leaf, that means we are going to pluck a leaf from each plant and check for the presence of starch. What do we think, that both the leaves show the presence of starch in the same amount? No, it will be seen only in B, not in A. Why? A will turn pale in color and B will definitely show a little amount of starch formation and will turn blue-black in color when undergone this iodine test, right? Because starch test means we have done iodine test. तो उसमें क्या होता है, A वाला पेल येलो रहेगा, B वाला हमारा स्टार्च की प्रेजेंस की वजह से थोड़ा सा ब्लू ब्लैक कलरेशन दिखाएगा. What does it conclude? That carbon dioxide is necessary for photosynthesis, sunlight is necessary for photosynthesis, right? Here carbon dioxide is absorbed by this KOH which is kept in this setup. जब CO2 ही नहीं मिलेगा plants को, then it will not show that formation of starch has taken place, meaning CO2 and sunlight both are required for the process of photosynthesis to take place, right? Moving next to एक छोटा सा टॉपिक, डेंटल केरीज. याद है डेंटल केरीज क्या होता है? हमने न्यूट्रिशन किया, हमने सब कुछ खाया, डाइजेशन भी किया. देन वी से दैट द रीजन ऑफ द tooth, we have our teeth fixed in a socket, and this soft region of tooth is normally known as enamel, the outer covering. This gradually gets soft. It begins to soft when the bacteria, it acts on the sugar which is mainly present in our food and remains sticking here onto the teeth. We say that when these small food particles stick to the teeth, they form dental plaque. Saliva cannot clear it off entirely from all these areas. Saliva नहीं clear कर सकता because sometimes it is not able to reach here, and it cannot neutralize the acid as the plaque covers the teeth. So brushing after eating, or we say before you go to bed during night time or in the morning, so it is regularly required. Why? Because after eating it will remove all the plaque and the debris, the bacteria, whatever is sticking here, and it will not produce acids. Bacteria will not produce acid, so no microorganism will invade the pulp. ये पल्प होती है ना हमारी, जो पल्प कैविटी, यहाँ पे ब्लड वेसल्स होती हैं, बहुत ज़्यादा पेन होता है आपको. तो इट इज़ ऑल बिकॉज ऑफ द इन्फ्लेमेशन एंड इन्फेक्शन इन द पल्प, and that occurs because of the bacteria which produces acids, and plaque formation automatically deteriorate the quality of enamel, which is here the covering of the tooth. So this is an important topic, like why do we have to take care of our teeth, and what happens when these food debris, they remain sticking, or food particles remain sticking to the teeth, because they lead to dental plaque. Thank you.